Yes, we can start. Hello, everyone, and welcome to this webinar on rapid approach to vaccine development. This webinar is part of a series of webinars with the goal of providing information on vaccine development and available career options in the field of vaccinology. My name is Clive Nyaribo, Assistant Research Officer at the KEMRI-Wellcome Trust Research Program, providing support to the Vaccinology Workforce Development Program, together with my colleagues, Professor George Warime, Marianne Munene, Caroline Mate, and Rinea Imbai. Welcome you to this session. Before we get started, a few housekeeping matters. Kindly take note that this webinar is being recorded. Kindly feel free to use that chat option to share your questions during the presentation, and we'll address them during the Q&A session. We'll have a few open slots for people to directly ask questions to the presenters. If your internet is stable, you can raise your hand using the available icon. As earlier mentioned, this is a series of planned webinars with the goal of providing information on available career options in the field of vaccinology. This being run under the KEMRI-Wellcome Trust Research Program, which is a renowned health research unit of excellence based in Kilifi and in Nairobi, as well as Mbale in Uganda. Over the years, we have excelled in the use of novel ideas and collaborations to achieve better health for Africa, while also developing African scientific leaders. Our vision of the program is to undertake cutting-edge and novel research relevant to national, regional, and global needs. It's under this program that we have the Vaccinology Workforce Development Program that aims to expand the technical know-how and capability for sustainable end-to-end vaccine research and development in Africa through training and knowledge exchange activities in the vaccine ecosystem. This involves areas such as target product profiling, research and discovery, preclinical and clinical studies, biomanufacturing, regulatory affairs, and deployment. The workforce development is through 3 channels. We have internships at human vaccine manufacturing institutions that aim at establishing mentorship scheme that links interns with senior vaccinologists and health market systems and engagement academics globally to share, strengthen experience and research experience in vaccine research and development and manufacture and enhance career development. We also have a fully funded 2-week master level course in vaccinology presented both in English and French. To successful applicants will be selected based on their direct involvement with vaccine research and their excellence in professional and academic achievement. And this year we have it happening in Dakar, Senegal. 3, we also have webinars like this one to hire career scientists, graduate and undergraduate university students in Africa to provide information on possible career options for university students for informed career making or career decision making. This program is an arrangement with the African Union's Partnership for African Vaccine Manufacture, PAFIM, ambition to increase local vaccine production to meet 60% of Africa's routine immunization needs by 2040. And strengthen the capability for vaccine research, discovery, and manufacture in Africa. And this webinar series is part of this engagement. Today, we are glad to have Dr. Kay Hunsberger, who will be discussing rapid approach to vaccine development. Dr. Kay Hunsberger is the Executive Director of Vaccine External Development, leading and connecting key activities with external strategic partners from Merck, Sharp and Doom, vaccine development and manufacturing, life cycle management, and pandemic readiness. As the strategic CMC liaison, our roles include fostering strong partnership and facilitating collaboration to drive scientific advancement and global access of vaccines and drugs, with a focus on low and middle-income countries. She also collaborates with MSD, internal stakeholders in discovery, clinical, commercial manufacturing, regulatory affairs, and legal to develop a robust plan for pandemic or epidemic preparedness. She is also the CMC representative for external collaborations with government agencies and NGOs for epidemic preparedness. Ms. Hunsberger's experience spans over 30 years in the development, technical transfer, and commercial manufacture of live and recombinant vaccine and biopharmaceutical products. She has also led the development effort for Ebola, and our team produced the emergency use supplies that supported Ebola outbreaks from 2016 to 2020. She most recently led an effort in partnership with in the International AIDS Vaccine Initiative to produce a Sudan vaccine for the recent outbreak in Africa. Across her career, she has been involved in the development and commercialization of most Merck vaccines. She received her PS in biology, biochemistry, and a PA in marine science from East Stroudsburg University. Over to you, Kay Hunsberger. Thank you. Welcome.
Thank you so much for the kind introduction, and thank you, Caroline and Clive, for inviting me to speak today. I just want to do a check. Everybody can hear me correctly?
Yes, we can.
Super, thank you. So today I'm here to speak about a rapid approach to vaccine development, and I welcome any question that you might have about, be it my career or about development of vaccines. And so today I'm going to talk about the approach to rapid response, how we approach product and process development, how we approach analytics, manufacturing, the regulatory approaches, and then I'll give 2 case studies, one for Ebola and one for COVID. And then continually we're learning lessons about how to improve, and then just acknowledgements to many of the people involved. So, if you don't know Merck very well, Hilleman was a key scientist here at Merck who helped deliver many of the vaccines that Merck has been involved in. And so across his career, he was able to impact, he had 40 plus vaccines in human and animal development as a part of his career. And I was lucky enough to actually work during some of his era here at Merck. And so not sure why the slides aren't advancing. So I wanted to just share, typical timelines to develop a vaccine are 8 to 12 years. And so at the time of the Ebola epidemic, right, people were dying and we needed to make a vaccine quickly. And so we had to look at a very different way of approach. And so things that we thought about as a part of this was first to think about what product technology would be appropriate to use. So, many of you have heard about the COVID outbreak and messenger RNA, right? But in messenger RNA, we haven't been able to generate memory response, right? Again, something that we'll continue to work on as scientists, but right now that's one of the challenges. If you think about viral vectors, some of the viral vectors have variable memory response. Some are very good, and I'm going to give you an example of a very good one, and then some aren't. And so, Ervepo, which is the Ebola vaccine, did great at generating memory response with single dose. But if you look at J&J's, it was a 2-dose regimen, right? So how do we develop vaccines so that a single dose is sufficient? And lastly, protein-based vaccines, again, you experience variable protection. So there's no single solution to developing vaccines. You really just have to look at all the different platforms and see what will give you the right response. And so relative to Merck, as a part of Ebola, we recognized that we needed to move to platform approaches. And what do platform approaches ultimately give you? You'll have strong technical knowledge in that platform, both process and analytical. You're able to leverage your pre-existing animal studies. You're able to stock materials, consumables, would allow you to start the laboratory work quickly and also to get into manufacturing rapidly. The great thing about a platform is you can align it to the manufacturing facility so that the facility is ready to take it, right, and be able to transform it into product quickly. So we intentionally use a lot of single-use technology, standard unit operations. Again, that allows for quick and rapid transfer, both from the clinical manufacturing facility into the commercial facility. And then depending on demand, we would look at the idea of implementing a continuous manufacturing process. So one of the things that's really important is that you have well-characterized vaccines. And so from a perspective, one, you must have its biological activity and understand, its potency, shall we say. And that can be done with many different assays, as noted here. Additionally, you have to understand its structure. You have to understand its purity, right? And what impurities are present, right? And minimizing impurities, whether you have heterogeneity, like in the case of our repo, defective viruses versus effective viruses, right? You need to understand the sequence of the product, and are you repeatedly getting the same product, right? And that it doesn't mutate, for instance, like a virus. And then additionally, doing secondary and tertiary structures such as, DLS or cryo-EM, those types of things. Lastly, very important to understand whether it aggregates and any subphysical particles around it. So it's important as you continue to think about the analytics of how do you bring speed to analytics, and that's transitioning to real-time release. So conventionally on the left-hand side of the slide, that you would typically initiate manufacturing, you pull a sample, it goes, chain of custody to a QC person, it moves to the lab and gets tested, results put into a system, and then, a review is performed and the data is approved, right? Now we're moving into real-time quality release, and the idea that a test is conducted right out on the floor, immediately the data is moved to an electronic system and then subsequently approved. And additionally, as a part of that, this idea of on the right-hand side, we're looking at, modeling your MVDA, your process, your systems biology to better understand the product itself, how it's manufactured, and what opportunities there are to improve the overall process. So end-to-end analytics are multimodal vaccines. So again, on to the left-hand side, I'm showing you the different types of vaccine: live attenuated viruses, genetically modified viruses, viral-like particles like HPV, polysaccharide conjugates. A great example of that is pneumococcal vaccine, right? Messenger RNA vaccines such as COVID, right? And then to the left-hand side, I'm showing you what do you need to do to develop the assays across the manufacturing development. And so when you first start in discovery, you're using analytical method development. You're trying to figure out how you can describe the molecule. And then you further develop that method in phase 1 to actually support release of that material into clinical trials. And then you subsequently continue to improve or enhance the assays to the point that you validated phase 3 and into commercial manufacturing. And then again, just in the table, some key attributes that are required. Again, we talked about this potency. You have to know what you're putting into the vaccine. Its identity, we tend to do sequencing or immunoplaque assays, understanding your impurities, and then the safety profile of the material. On the bottom part of the slide, I'm really showing you an opportunity of how you can innovate. So when we first developed the plaque reduction neutralization test, or PRNT, for Ebola, it was first you seed cells, then you dilute into an assay plate at day 2, and it took 8 days to make a read. We were able to innovate across that assay to alter it so that we could actually get readout in 3 days, right? And this is really important because it allows the speed of the clinical trial. We also, as a part of that development, were able to lower the cost and higher throughput. From a manufacturing perspective, it's important that it's a self-supporting manufacturing facility. So here at Merck, we have a clinical manufacturing facility that is capable of actually manufacturing 3 products at the same time, right? So it allows us speed into manufacturing. And again, we focus on the platform processes. So in this plant that we have here, we have over 1,200 pieces of equipment. So we're ready to manufacture any different process, any different delivery of structure, shall I say. There's no cell line or bacterial yeast mold that this pilot plant can handle, right? Staff are trained to run all these process operations so that they're quickly able to respond. And then we take a risk approach to the manufacturing schedule, and I'll give you some examples of that when I talk about Ebola. So again, this clinical facility is flexible. It's able to do non-GMP or development work and also to do GMP work. And again, when I talk about these different suites, one suite could be doing development work and another suite could be in GMP manufacturing. It is a combination of single-use and stainless steel because some of the platforms require stainless steel. And it has an open floor concept with the idea of rolling in equipment. And here's just a diagram or a visual that gives you an idea. So in the center, you see a footprint of a layout of kind of a large room where equipment would be rolled in to support a manufacturing process. Again, we try to use single-use closed system processing because that brings rapid speed and to the development and easy transfer to other plants, right? Then the idea of this the flexible multi-product facility, you see the layout of 3 different suites, and the 3 different suites having different kind of mirror platform open concepts, but might have some different capabilities, i.e. stainless steel in one suite, single use in another suite, maybe alcohol feed, some different capabilities. So again, being able to provide latitude to make anything we need. And then we have quite a bit of automation to support the continuous processing. And as we noted earlier, that the ability to do analytics right in the suite. So we actually have a little area where equipment are set up to do analytics right as product is being manufactured. From a regulatory approach, right, things are changing. And certainly from Ebola to COVID, major changes in regulatory approaches that are continuing to grow. As I say, you could see the regulatory agencies recognizing the need for change. And so many countries provide alternative regulatory approaches to diagnose, treat, and prevent disease. And so the first is the use of the animal role. And so, you know, the idea that you could do, if you have a good animal model, that you could limit your amount of clinical testing and again, move through the development process rapidly. The utilization of prior research work. And we've done this in our partnership with Ebola. And then when we worked on the Sudan vaccine, we utilized the prior research work of the Zaire vaccine, right, to support the Sudan vaccine with IAVI. And that allowed us to get into the clinic much faster. Then also having intermediate clinical endpoints. And again, in the Ebola program, I'll give you an example of how we had an intermediate clinical endpoint that allowed us to make reads quickly and move forward in development. What's really important during the development of a product like this, when you're trying to develop with speed, is enhanced dialogue with regulatory agencies, constantly having conversations with them, making sure they understand what you're doing, what you're trying to deliver, you know, and making sure they're aligned with the approach that we're taking. And so as we developed our key clinical data, we continued to report them into the agencies and rapidly progress phase 2 to phase 3. And again, in Ebola, we had 23 informal and formal regulatory CMC interactions, which is, you know, extensive. At that time, it was unheard of, right? And then, of course, we were the first to file the emergency use authorization listing, which occurred in May 30th of 2016. However, it's important to note that while they had this emergency use authorization listing, the regulatory agencies didn't know how to do it. They had never done it before. And so there was a realization that they were trying to understand. And so ultimately, we provided the doses under a pre-licensed patient access clinical protocol, which is the idea of making sure that a compassionate use protocol. And so now I'm going to talk a little bit about our VEPO and the actual efforts that we went to. So to kind of describe, this was an experience of a lifetime for me. At this point in time in my career, I had developed many vaccines, but what had happened was a small company called New Link Genetics reached out to the FDA, and the FDA came to Merck and asked us to partner with New Link Genetics to help them bring forward the Ebola vaccine. And it was an unprecedented experience. It was the first time that there were public and private sector all getting together to make a difference and to get this vaccine into country and to the patients, right? Because again, many people were dying and people were very ill. And so we obviously partnered with the African governments and researchers and volunteers out in country. The product originally started at the Public Health Agency of Canada, right? And then was developed in the United States and in Europe. And then Merck helped bring it along, which is in the United States. From a field response, there was many service organizations that were helping down on the floor: Wellcome Trust, the Gates Foundation, the Doctors Without Borders, Red Cross, etc. Everybody was doing their part to help bring this forward quickly. So what is VEPO? What essentially it is a genetically modified recombinant vesicular stomatitis virus, which is we refer to as VSV. And so in the left-hand side, you see the structure of the virus itself. And all we did was delete the glycoprotein VSVG, glycoprotein, and we substituted the Zaire glycoprotein in the structure and then replicated that virus. And so it's not a live virus, it is a genetically modified virus, right? So again, somebody wasn't going to get infected from this and get disease. What's really important is to talk about what went on during that time. So there's a huge outbreak going on. And as we purchased the company and the materials, there was a realization that there was very little supplies. There was 150 to 170,000 doses. So what we had to do is make decisions really rapidly on how we were going to get that to the patient. And one of the things we actually did was an expanded ring vaccination trial, which hadn't occurred in years, right? And it was actually a process that we used to make sure that we were getting to the patient and the close community contacts, right? And making sure that we were closing down the spread of the vaccine. The second major effort was to ramp up our clinical manufacturing facility, which I ran, and to produce additional doses as quickly as we could, right? Because 170,000 doses was very little. And at this point in time, by the way, we didn't know how much we needed. And that was, very interesting too, who FDA, EMA, working with Merck to kind of help us decide how much volume we need to get out there. And everybody had a different opinion. But anyhow, essentially, we felt it was an ethical obligation to get vaccine available as rapidly as we could. And so we utilized the WHO emergency use authorization process to drive the manufacturer. Lastly, our second objective was to make sure that we could get this product licensed quickly. That means we had to be able to get up a manufacturing facility, right? Demonstrate safety and clinical benefit, right? And then move to PPQ activities and file, right? So we have 3 different distinct initiatives going on, right? First one is really the clinical, make sure we have clinical. Second one, making sure that we're making more clinical supplies. And lastly, moving it into commercial. So what happened? So we were reached out to in September, right? And in actually 5 days, we signed agreements, which again was unheard of, right? And we actually start a phase 1 clinical trial October the 13th. So in under 6 weeks, we were able to start a clinical trial, right? And then we actually were able to select dose from that result in phase 1 trial by January, right? I apologize there. So as you can see, in January, at the end of January, we select dose. By the beginning of February, March, and April, we start 3 clinical trials. We start a phase 2/3 PREVAIL trial in Liberia. We initiate a WHO phase 3 trial in Guinea. And we initiate a CDC STRIVE trial, which is a phase 3 trial in Sierra Leone. Again, now this outbreak is in multiple countries, and we're trying as rapidly as possible to get into those countries, to get vaccine to the people, and to get the clinical data. We were able to actually do an interim need analysis, right, of in July, and actually demonstrate vaccine efficacy. And so that article was presented in Lancet. From then, as I said to you, as soon as we had clinical, the next thing we needed to do was to initiate the consistency trial because we're moving to commercial, select the commercial siting, right, and initiate CapEx. So you can see in August, we initiate that phase 3 safety lot consistency trial using material that some of that 170K doses that was existing. We select the commercial siting and we initiate a CapEx project to be able to make the product at that site. And we also filled all the rest of all the remaining drug substance into drug product in November. From there, this is where the plant gets involved. In May of 2015, we initiate the manufacture of the banks and the seeds to support the drug substance and drug product manufacturer. By December, we have completely developed the manufacturing process, and we initiate manufacture of drug substance. And in January, February, we manufacture this drug product. And so how do we go about doing this? So one of the things it was recognized was we don't have much time to develop this product, and we had to do it, and our goal was to develop the process in 3 months. It actually took us 4. But what we said was, what do we know from the literature? What do we know from our own expertise? And what do we know from the CMO that had this product previously? And what we did was we said, the cell expansion process is Vero, right? And Merck has many products in Vero. And so we said, we're not going to do anything in the cell expansion process. We're going to strictly look at the infection of the virus, when to harvest, and look at the downstream operations, which was depth filtration, enzyme reaction, and TFF, right? Our process development kickoff was in June of 2015. We actually shipped the bank and the virus to us in May. So it gives you perspective. From May, we get the material. We're already running experiments by June, and we complete all the scale-up experiments by October, right? Subsequently, end to end, it was 1 year to produce drug substance sufficient to make greater than 750,000 doses. We produced greater than 125,000 doses in a 10-dose image. We filed the EUAL, right? And then we had doses available by 30 September of 2016. And I think what's really remarkable too about this is, remember that while we were developing, and I'll show you a slide on this, things are changing. We're getting data and assays are being developed at the same time. And so this slide is really a reflection of that. And if you look at the graph on to the left-hand side and harvest titer, right? You first have the working virus seed and the GMP working virus seed that we had developed versus the CMO. And you can see that we have variable data in comparison to the CMO experience, right? And so what about the process were we doing differently? How do we make changes? And then you can see that we started to get comparable results in the 400 roller bottle process. But we also additionally did some enhancement work. And so you can see across the development scale-up work of 90 roller bottles and into the scale-up of 400 roller bottle that we were able to enhance the productivity of the process too and get more increased output. And in reflection across the opposite sides, just showing it graphically how we were able to prove the overall yield of virus. And let me say to you that the virus was there. It was how do we make sure we release it from the cell debris and collect it and keep it viable, right? It's not as if we really produced more virus. We enhanced its ability to harvest it, right? And so if you look at this graph, all the black represents all of the data coming out of the CMO, right? And then the blue was the experience of the GMP lots at the CMO, right? And red are the experience of the Merck lots. And you can see that they get the higher amount of productivity as in comparison to the work done at the CMO. So what did the process look like, right? Ultimately, we had a single-use process that was manufactured in roller bottles, right? We harvested and then we clarified through a clarification filter. Then we did what is called a nucleus digestion and enzyme treatment. We ultrafiltered and then we dispensed and stored. And it was a single use with a fully closed downstream process. Right. And I can say to you, we continue to develop, we enhance this process, and we now have a fully closed process. It only has 2 aseptic manipulations in that process as a development we're going through right now. Right. From a drug product process development, what did we have to do? Because, one, we need to make sure that we could create a drug product that could be brought out into the field and could be stable. So we looked at impact of shear and mixing on the final formulated bulk. We actually looked at drug product freezing and thawing, its dilutability to make sure it had linear performance, and short-term stability, right? Its stability at 2 to 8, because we wanted to understand how much time we had in the field to vaccinate people, right? And then understand what we call time out of refrigeration, which is the open spots in the manufacturing process where you can't keep it cold, right? The room temperature effects and how they impact the potency of the virus, right? What did we do to make sure about analytical comparability? If you remember, there was a contract manufacturer that was supporting it. This contract manufacturer was located in Germany, right? And they had the process. So we had to do in the biologics pilot plant, which is the plant that I was leading, we had to do an analytical comparability from the contract manufacturer, the biologics pilot plant. Then we had to do an analytical comparability from the pilot plant to commercial facility. And then subsequently, we continued to use that same analytical comparability for any manufacturing changes that we make to the process today. Right? It's important to kind of talk about the regulatory strategy here because it was very complicated, right? Prior to this, it's agencies had their own way of doing things. EMA doesn't do it the same way as the FDA, and certainly WHO qualification has their process, and of course each African country has their own process. And so the complexity associated to getting regulatory approvals was extremely complicated. And so some of the challenges were the lack of harmonization, right? Which we understood, right? But understood that we had to meet both the FDA's requirements, the European requirements, WHO's requirements, and the African country requirements. Of each African country, we were looking to get approval. And today, this product is approved in 14 African countries. So it was a unique situation that we were developing a vaccine during an outbreak, right? Versus that standard timeline I showed you of 8 to 12 years. Like just no longer acceptable today. And I will say to you, inside of Merck, once we did this Ebola vaccine, it set a new standard inside of Merck of how quickly to develop vaccines, right? Lastly, high risk of vaccine to produce economically. Like when you develop with speed, sometimes you forfeit cost elements and then have to, and so ultimately that's what's happened. We've gone back, right, to kind of do things to lower the overall cost of the vaccine, right? What happened? We actually produced a single dose image stored at -70 that had short-term 2 weeks worth of storage out in the field, right? And this is actually something we're further working now on to try and extend time out in the field. But as I shared with you earlier, we were able to develop the product in a single year. We had a single-use technology that we were able to rapidly transfer to the commercial facility. We filed the EUAL, even though we didn't ultimately use it, right? We were able to get doses into the field by September, and so we were rapidly able to begin to affect people in the country. And what was really important was we were able to support outbreaks for more than 4 years out of the pilot plant. And something our team was very, very proud of. There's posters all over our building about Orvepo and the Orvepo experience, right? And we got approval in the US in December of 2019 and in Europe in February of 2020. Now, the COVID vaccine. And so, now that we have another outbreak, right? And can we leverage our platform and manufacture in a simpler way? And so this was a partnership of Merck with IAVI, and we looked at the VSV construct, right? Again, with this idea of changing out the glycoprotein and substituting in the COVID virus, right? Using the VSV. Some challenges that are really important to note that you can see on the left-hand side of the graph is that, first of all, these viruses are from different virus families, and the replications of the way they replicate the virus vary considerably. And so there were concerns, one, sheer size, right? That the COVID molecule spike protein was much larger, right, than the Ebola virus, right? Additionally, the viruses replicate in 2 different cellular compartments, right? And then they concentrate in different regions of the cell after being replicated, right? But anyhow, what we did is not any different than we talked about earlier. We took the VSV construct, where it is red G glycoprotein, number 4. We spliced or deleted that out. And just like we did in Zaire, we now put in the spike protein, right? And try to produce a product. And I think what's really important here is now, will the virus, a different virus, behave the same way in the manufacturing process, right? Are you going to do it the same way? Well, here was our learning opportunity. From the time that we did Orvepo, we had developed another platform, a microcarrier platform, so we could put it into a bioreactor with glass beads, have the cells attached to the glass beads and replicate, and then bring in virus and infect. So we changed the upstream operation. From a downstream operation, it was fairly close to these unit operations, but not identical to Orvepo. And this is something about a platform that while the platform can work, there's probably changes that have to be made to support the particular virus you're working on. And so, as we said, we had a commercially ready microcarrier process that we were able to implement in 3.5 months. So if you remember now, it was 4 months to do the development process with roller bottles. We had this microcarrier process, and I would say to you, it was more of a scale issue. We had to go up to 2,000 liters for COVID because it was a worldwide outbreak, and so that's what made it 3.5 months, a little longer in manufacturing time, right? So therefore, harder to do the development. So this process used cell stacks going into an N-1 bioreactor, and then it's the final bioreactor. The virus propagation is done in that bioreactor. Then the nucleus digestion, where the enzyme treatment that we talked about previously was moved forward into the bioreactor as an opportunity, right? Then we clarified with a filter. We had to add a column chromatography. And the reason why is at the 2,000 liter scale, there's way more cells. So we had more host cell protein that we needed to remove, right? And so we had to put in a column chromatography, then the ultrafiltration. And then this time we implemented a sterile filtration so that we could eliminate closed system processing. And this makes a huge difference in how you manage the facility and your facility costs. Again, and facility costs directly influence COGS. So now your price per dose can go down, right? Which is the goal. So how do we move with more speed here? So now, if you remember at the SARS outbreak, and if I think about the SARS outbreak in our plant, right? Merck told people March 11th, you were stay home, don't come in, right? And my team was told you have to come in every day, right? Because we actually had 2 COVID vaccine products that we were working on. And so in under 5 weeks, we selected the clone. And when I say selected the clone, that means including developing of the clones and sequencing of the clones and looking at productivity of the clones. So in under 5 weeks, we did all that work. And we moved forward with the clone that we selected into process development. By June, which is only 2.5 months, we manufactured the master virus seed. And as you can see from an overlapping process perspective, we began cell expansion while we were making the master virus seed. So that we had to get this potency out. This is actually where we partnered with an outside testing firm, and we had them put on extra plates and everything to ensure we got the data out so that we could actually infect the drug substance and then harvest it and get drug product into filling. And we were able to do that in August, right? And well, what's unfortunate is while the drug development team, our team was doing a great job in CMC, we had delays in the clinical trial design, right? And so it took more than another 2.5 months to get through the clinical development design protocol, IRB approvals, so that we could do first in human by November. But while that clinical study was going on, we immediately went forward into our phase 2-3 process development. We manufactured our GMP material to go into a phase 3 clinical trial. However, if you're not aware, we didn't get enough memory response. And in my opinion, a big part of this, I think, is because of the challenges I shared with you relative to the development of the virus and the fact that the virus replicated in different places and actually stored in different places in the cell. And I think that ultimately had an influence on our ability to collect the virus productivity and then to create memory response, right? And so what lessons have we learned, right? That platform processes and standard unit operations are the way to go because the equipment's there, I can roll it into the suite, and I can quickly move forward and develop a product. One of the other things we did was early integration with discovery. Like discovery and the plants worked directly together on the clone selection so that the plant was ready to manufacture rapidly behind them. Minimizing repeat scientific work, right? Making reasonable risk-based decisions in your development processes. And then, of course, front-loading analytical resources because you needed to understand the analytics of the product, right? From a manufacturing perspective, use existing facilities to support PPQ and address long-term capacity off critical paths. So this idea that now could you use a facility inside of your company that produces initial PPQ lots while a commercial facility is actually being built off critical path. That was another opportunity at speed. Designing and implementing these multi-purpose flexible facilities and making sure that these facilities exist end to end, right? that from a regulatory perspective, understanding the manufacturing ability, the timelines, etc. The technical reports that we did rolling submissions to get to licensure quickly and to partner with the agencies. And just to remember that everybody is unique from a regulatory perspective, right? And of course, the emergency use authorization, right? These processes were scientifically unprecedented, right? It was unprecedented partnership. Merck, it was an awesome experience to work with all the agencies and Doctors Without Borders and, people down in the field, et cetera, in order to deliver this product into the African countries, right? And the product itself was unprecedented. And again, the idea that we're so many partnerships, whereas in the past, and again, this continued expanding, COVID didn't. And I show you this picture because this young little girl, her mother got a dose of the vaccine. She was one of the first people and was protected. And so she sent this picture, thank you, science. And so I hung this picture in my plant to remind people of who we were trying to make this product for and our importance of delivering a difference. And before we start questions, I just wanted to acknowledge all the various partnerships that occurred across the project and all the different agencies inside of Merck, governments, et cetera, nonprofits, in order to deliver this product. Thank you. And I'm happy to answer questions.
Thank you. Thank you for the insightful presentation. I'll move to the questions on the chat.
Do you want me to stop sharing so I can see the chat?
Still okay. They might refer to the presentation.
Okay. I'll leave it up.
But you are not able to see the chats?
No. So
okay.
That's okay. So I'll just rely on you to say the questions.
Okay. Okay. Let me go to the questions that have been asked on the chat. It will not be in a particular order. I'll start from the first questions that I see. Anton is asking, did you encounter some difficulty of conflict of interests in the process, or how are you protected so that you are the only people organization being mandated to manufacture vaccines? He's asking about the mandate to manufacture vaccines. Kindly go ahead.
Yeah, so I would say no in this case. I actually would share with you that during the time of the Ebola outbreak, right, we actually partnered with several of the vendors too. We actually told the vendors that we had an opportunity to make this Ebola vaccine and to get it into Africa quickly. And so we went to a couple key vendors and asked them to support us in getting key supplies rapidly so that, essentially they bumped people in line to give us, media and bags and etc. And we were very clear that, hey, you can use it as an advertisement, etc. But here is your opportunity too to make a difference. And so I will tell you that, this was probably, again, the highlight of my career because there was such great partnership and this idea of conflict of interest didn't arise. Right. And I will tell you, pharma is a competitive industry, right? And we don't necessarily share our secrets. But that was not the case in this question. It was like, we need to do this and we need to do this now.
Okay, very nice. And then there's a question from Godwin Ngeno. The question is, what is the immunity duration of the mRNA vaccines? He's saying that because we all know that these vaccines may wane over time and mRNA live for a short period. The immunity duration for mRNA vaccines.
Yeah, so I think if you're not aware, essentially we're seeing that the messenger RNA vaccines are dissipating anywhere from 6 to 9 months, right? And so there's a recommendation to vaccinate yearly for COVID, right? And that is exactly that. What has happened is we haven't triggered the memory response, which are the B cells, right? We have triggered a response, an antigenic response, but it's a very short-term antigenic response. And that's something researchers are working on now, right? Because if we solve that problem, right, messenger RNA definitely is speed. Like you can develop and bring a messenger RNA vaccine really rapidly, and in the manufacturing process is even faster. Like it's a 2-week manufacturing process, and so you could, from a speed perspective, definitely deliver faster than a vaccine. But what we haven't done is solve the memory response. So that's why you'll continue to see, I think, both being developed, right? And scientists will continue to look at how do we have the B cells be recognizing that messenger RNA so that we gain memory response.
Thank you. I'll go to the next question by Mkanda Kadima. He's saying, Dr. Hansberg K, I'm so impressed by the work on vaccines that you have been involved in for years. This makes me interested in vaccine development. I could greatly appreciate your guidance and orientation for those willing to build their careers in the development of vaccines.
You know, that is great. You know what I'm going to do? I want first, I don't remember if my slide has my email, but maybe I'll partner with Clive, with you and Caroline, that we can share my email. I'm happy to connect with people, have conversations. I'm very much about people's career development and would love to, talk with anyone about what I could do and guide people in their careers. I do it quite a bit inside of Merck. And I also do it with colleges and universities, and I even go speak in middle schools and high schools. And so I'm happy to talk to anyone online here. And so we'll make sure that people have my email to reach out to me.
Thank you. That's
In
very
fact,
kind of
right
you.
now we haven't had the need to show my slides. So why don't I, right here in the chat,
Oh, okay,
right?
fine.
I'm going to right here in the chat put my email.
Okay. I'll go to the next question. This was asked earlier on. Godwin Ngeno says, good afternoon, everyone. I'm Godwin Ngeno from Masinda Murio University. I'm partnered on this field of vaccinology. Are there any platforms to help me get sponsorship to get hands-on experience on vaccine development? More or less the same like the other one. You can go ahead, Kendra.
Yeah, the how is always a difficulty, right? I'm aware of the KEMRI program and the fact that you guys have a development program where people can do internships, etc., right? And that they can get experience. Certainly, Payan and Eryani Hamid, who's on, I think, online. Hillman Labs also is doing a partnership with KEMRI-Wellcome on giving apprenticeships for a year at Hillman Labs in Singapore that people can apply for, right? And then, again, continuing to get your resumes out to people across these both nonprofits, the universities, and pharma, because we do want to sponsor the opportunity to help deliver experiences into the African countries, for sure.
Thank you. Also, Caroline will chip into that question.
Yeah, thank you for that. That was a very good question. So right now, there's a very big drive to increase the workforce development within the vaccinology space, and KEMRI-Wellcome Trust is running what we have as the 3 pathways that my colleague Collins had mentioned about. So one is using the webinars such as this just to pass on information to a very wide range of interested people. Then also based on that, then we also have the vaccinology work, the apprenticeships that we do in partnerships with various institutions such as Hilleman Laboratories, where we place shortlisted candidates within these institutions to get hands-on practical information and learnings, and it's anything from 4 months to 1 year. And these are highly competitive, I have to admit that, since they are limited capacities and they're fully funded. The third opportunity that we use is the just bringing in scientists and researchers and people from the entire vaccinology pipeline within an institution for about a week or two and run a course where it's very intense, kind of running through what the vaccinology research, development, and manufacture pipeline looks like. So that's what we're doing as Wellcome Trust. I know CSIR in South Africa also has a similar program. And also within the West African bloc, there's IPD, and also I think Egypt is interested in running something like that. So it's just a matter of keeping your eyes open on these opportunities and the call for applications when they come out, and building the networks with other vaccinologists so that when the opportunity comes in, you're looped in. I hope that has been helpful. Thank you.
Thanks, Caroline.
Yeah, I'll go to the next question from Scholar Kamwatea. How did you settle with the decision that the COVID vaccines will be injectable vaccines and not orally administered? Also, according to CT modules, it's clearly stated that a vaccine should take up to 12 years of manufacture process. How did you measure the safety of the COVID vaccines within the short period of less than a year? Thank you.
Yeah, so this is really associated to technology getting better and better. You know, if we look at when we take about that 8 to 12 years, it's kind of looking back at rotavirus or something that was 20 years ago. And so, you know, we now have PCR technology. We now have the ability to transcribe and translate proteins and rapidly express them in different types of formats, be it a yeast, be it E. coli, which is a bacteria, or doing mammalian cell culture, etc. And so I think it's just science continues to get better and better and faster. And so again, there are some things you can't bring speed to, and I'm going to use the animal models, right? Like an animal model takes the time it takes to run an animal model, right? And so I can't shorten that down. It's, if I do an NHP study, it's 48 days, and it's going to be 48 days. There's not anything I can do about that. But what I can do is look at all the things in front of that to get into the animal model quicker and get that data back out quicker. And I could be doing development work or doing things while the animal studies are going on to move it forward, assuming I get good results. Right? That's kind of the decision you're making. You're making risk-based decisions to move forward, assuming you will get good results, right? And that's what's making a difference. Whereas before, when you did the 8 to 12 years, you first did the discovery work, and then you put it into animal models, and then you waited for the animal models to get the results off, and then you did the next step. That's no longer happening. It's not sequential anymore. Everything's happening in parallel. Right? And again, speed and science. The technologies are getting better and better. And I use a great example. You can do mycoplasma release testing, which takes 30 days, or you can do PCR, which takes 14, right? So there are examples of where technology keeps improving and bringing speed. And it will continue to keep happening. Like science will continue to progress. And some of you, I'm sure are much younger than me, right, are going to see that change across your career like I did. And it's going to get even quicker.
Yes, for sure. Thank you for the answer. So I'll go to the next two questions. I'll combine them, talking about career also. There is one, Emmanuel Pius, saying, Clive, you at the beginning, you said that there is scholarship basis on the vaccine. I'm not aware on that country help. Then there is another one. I'm passionate about pursuing career in vaccinology, currently in Nigeria, so no university is offering solely master's degree in vaccinology. Please do how do I navigate? Maybe Kay, you could be able to answer this if the masters in vaccinology and any other postgraduate degrees that can really align to vaccine development.
Yeah, absolutely. So I'm going to say to you, degree ultimately in vaccinology doesn't matter, because I'm going to tell you, you guys, my degree is in biochemistry, right? And so I actually knew that I wanted to work in pharma, right? And just happened to land my first job, at a small pharma company, actually working in the testing labs. But then in my second job, I got into the vaccine development, right? And actually was first for animals, right? And actually, how could I, affect crops, and how could I, health and welfare of animals, right? And then I went to GSK and now worked on human vaccines and animal vaccines, because they did both. And at Merck also does human and animal vaccines. And so I actually got my vaccine experience in my career path, not actually as an education. And so don't let that degree hold you back. It's more seeking the opportunity in pharma. And then one of the things I will say about pharma is great about doing is actually giving you different opportunities and doing different roles inside of pharma, which allows you to gain that knowledge and experience. Yeah, so my degree is in biochemistry.
Hello.
And does KEMRI want to say anything here?
Yeah, I can add on to that. You're very right in mentioning that there's no direct career line into vaccinology space, because again, I remember, as you mentioned, this is an entire pipeline from the ideation of what vaccine we're going to build, the molecules, the processes, the lab, the clinical trials, the community aspect to it. So the different ways of skinning this cat. So, for example, I'm a microbiologist, and I'm still within the vaccinology space. So it's, as long as you're within the health pipeline, you find your way as you build your career into the vaccinology space. It's just looking out for those opportunities and being deliberate about applying for them, so that then now you build your career towards that. Thank you.
And Caroline, I think that's an important comment, that you direct your career, right? And so you have to be intentional. I agree with that statement.
Thank you. Very insightful. I'll go to the next question from the Q&A. From your perspective, VSV are much applicable to which virus families of vaccine development?
I'm sorry. What was the first part of that question of vaccine development?
From your perspective, VSV are much applicable to which virus
Okay.
families? Yes, virus families for vaccine development. Yes.
Yeah, so that's a great question. And so I'm going to tell you that the VSV construct is just one of many, right? And so I do think that as you can see, like COVID, like we weren't successful COVID. You can't just put anything into the VSV construct and make it work. But there are families of viruses that are very amenable to it. I can tell you that within Merck, one of the things we actually did was we plugged and chugged many of the different families of viruses into the construct to see whether we could enhance or improve its productivity, right? And understanding what viruses it could handle and what viruses it can't. And so that's something about a platform that you need to do. Like, you need to kind of say, how will this platform work? and what are the dynamics or the range in which I can use this platform, and when do I have to alter it and go to another direction, right? And so, you know, if there's something that's glycosylated, then you're going to have to think about mammalian cells, right? Like it depends on what you need to produce, what is the right direction to take. Right. And I will say this to you too, by the way, because when I was at GSK, we used to have 3 teams, and the 3 teams would actually compete against each other to bring a product through the pipeline. So if a new product A, and I'll use one that we worked on, malaria, right? One group did it in E. coli, another group did it with mammalian cell culture, another group did it with something else, right? Yeast, maybe, Saccharomyces, and put them into the platform and would see which one was most applicable. And so a lot of that early development work is doing just that, throwing it into different platforms and seeing which one is probably the most applicable. And obviously doing a little literature work in advance of that, because there's some things that the literature is going to tell you that will eliminate some things and direct you towards others.
Yeah, it was great. I'll also check the chats and see if there is another question from the audience. If there is anyone with a question, can raise your hand. No another question on the chat. Okay, kindly provide your closing remarks, even for this presentation, kindly.
Sure. You know, again, to Caroline and Clive, thank you so much for inviting me to speak to the group today. I just want to say thank you for having me, right? To all the people on the line, you know, I generally mean what I said. Like I did put my email in the chat. Please feel free to reach out to me, and I'm happy to kind of set up time, right, with you. And so I do crazy hours. I'm actually working on a project right now with a partner with the Hilleman Labs in Singapore, and I'm working with a team in South Korea. So I'm all over the chart relative to meetings. So I don't want you to think that because you're in a country that's off cycle to me, that we won't figure out how we can talk. We will. I will make sure that I find time to meet with you. All right. So I do want to just say thank you so much. And I appreciate having the opportunity to talk with you all and share my story.
Thank you, Kay, and thank you very much to everyone in this webinar. This was very informative and interactive. Have a good day and looking forward to more engagements with you. Before we close, kindly requesting that you take a few minutes to respond to the post-webinar survey posted in the chat. There is a link for you to do the survey. We would like to hear about your experiences and what else you could like us to discuss in upcoming forums. Have a good day and looking forward to more engagements with you. Thank you. Have a good day.
Thank you.